Welcome to the deep dive. Today, our mission is really to cut straight through some pretty complex molecular genetics. We want to map out the exact method scientists use to find those tiny DNA regions that control complex traits.
Right, taking the guesswork out of it.
Exactly, turning months, maybe even years of fieldwork into, well, almost instant knowledge, predictive knowledge.
And this whole process, fundamentally, it's called genetic mapping. It's what enables one of the most powerful tools we have now in agricultural science, marker-assisted selection, or MAS.
MAS. That sounds like the real prize here, the practical application.
It really is.
So instead of waiting for, say, a plant to grow up and show you if it resists a disease.
Right, waiting for the phenotype.
You use these molecular markers, which are linked to that good trait, to pick the winners much, much faster. And cheaper, too, I assume.
Definitely cheaper in the long run. You're selecting based on the DNA signature before the trait even shows up.
Like selecting for that orange color in sweet potatoes, the biofortification aspect, just by looking at a DNA test result.
Precisely. It revolutionizes the speed of breeding.
So what's the magic behind that prediction? What makes it possible?
Well, it hinges on a really critical, though kind of invisible, mechanism. It's called linkage disequilibrium, or LD.
LD, okay. We hear that term a lot. Why is this non-random connection between alleles at different spots, the fact they sort of stick together, why is that so vital?
It's crucial because if the marker you can easily detect and the gene you actually care about are associated, if they're in disequilibrium, you can use that easy-to-find marker as a stand-in, a proxy for the gene itself.
Makes sense. And the main reason they stick together is just because they're neighbors, physically close on the chromosome.
That's the primary driver, yeah.
Yeah.
Genetic linkage, physical proximity, that's number one.
But looking at our sources, it seems LD isn't just about how close they are physically. What else keeps that association strong?
Right, you have to think bigger about the population's history. Things like population structure, any selection pressures that have been acting on those traits.
Like breeders selecting for something specific over time?
Exactly. Or even natural selection. Plus, genetic drift, those random fluctuations. Or major bottleneck events where the population size crashed.
Wiping out certain combinations?
Precisely. And even gene flow between populations. All these things can influence how tightly alleles are linked across generations, sometimes even if they aren't right next door on the chromosome.
Okay, wait. If the whole system relies on this linkage, but every time organisms reproduce, you get recombination during meiosis, which tries to break those links apart. How fast does our predictive power fade? Does the LD just disappear?
It decays. It decreases, decays, you could say, over generations. And it's a function of something called the recombination fraction, usually symbolized as theta.
Theta. Okay. So that's the probability that recombination happens between the marker and the gene.
Exactly. The higher that probability, the faster LD decays.
Yeah.
Which means in species with high recombination rates, you need your markers packed much more closely together to keep that prediction accurate.
That really highlights the efficiency gain from MAS then, if you get it right.
Oh, hugely.
There's a great example in the material. If you've got a marker linked tightly, say only a 5% recombination distance from your target gene.
Yeah.
Using that marker lets a breeder pick out 950 resistant plants from 1,000.
That's a fantastic success rate.
Now compare that to the old way. Just looking at phenotypes in a standard F2 population for a simple recessive trait, you'd only find maybe 250 resistant ones out of that same 1,000.
Wow. So nearly 4 times the efficiency.
Yeah. Think of the savings in time, land, resources, not having to grow out and test all those extra plants.
It's a game changer for breeding programs. But getting to that point requires a significant upfront investment. You need the right raw material.
You mean the mapping population itself?
Absolutely.
Yeah.
The mapping population isn't just any group of individuals. It's usually hundreds, sometimes thousands, created from a very specific controlled cross.
A cross between?
Between 2 carefully chosen parents. Parents that are really different, highly contrasting for this trait you're interested in.
Why the big contrast?
You need that contrast to make sure the trait and the genes controlling it are properly segregating, dividing up among the offspring.
Right.
You need genetic variation to map anything.
And the size of this population is critical, right? The sources mention N100 to 250 only gives you a skeletal map.
Yeah, just the basic outline. If you want really precise, fine mapping, zeroing in on the gene's exact location, you often need populations well over 1,000 individuals. It takes numbers.
And the type of population you create depends on how the plant reproduces.
Fundamentally, yes, because the goal is often to create something stable and repeatable. something you can test again and again.
Okay, like inbred species, things like rice, beans, soybeans, crops that mostly self-pollinate.
Right, the autogamous species, selfing over 95%. With these, you can generate stable lines.
And this is where things like recombinant inbred lines, RILs, come in.
Exactly. RILs are sort of the gold standard for many mapping studies in these species.
How are they made?
You start from an F2 generation, from that initial cross, and then you just keep self-pollinating individuals for many generations, maybe 6 to 8 or more.
Until they're almost completely homozygous, fixed at each gene.
Precisely. Each RIL is essentially a unique, fixed mosaic of the 2 original parental chromosomes.
And the big advantage is
Stability and immortality, essentially. Once you have a set of RLs, you can multiply them indefinitely through seed. They stay genetically the same.
which must be amazing for experiments.
It is. You can test the exact same genetic lines across different years, different locations, different environmental conditions. It lets you dissect how genes interact with the environment, the GXE interaction.
Okay, so that's the relatively straightforward case. What about the tricky ones, the outbred species?
Ah, yes. The allogamous crops. Things like maize, most fruit trees, forest trees like eucalyptus or pine. These typically self-pollinate less than 5% of the time.
And they cause mapping headaches.
They really can, for several reasons. Many have long juvenile periods. Think how long it takes an oak tree to reproduce.
Right, decades maybe.
Exactly. Crossing takes forever. Many also have complex genomes, high ploidy levels like potato or sugarcane. They're autopolyploids with multiple chromosome sets. That complicates the genetics and the statistics enormously.
And you can't just force them into RILs.
Generally, no. Many suffer from severe inbreeding depression. They lose vigor or fertility if you try to self them repeatedly. Plus, some have self-incompatibility systems preventing selfing altogether.
So geneticists need different strategies for them.
Much more creative designs, yeah. Things like full SIB or half SIB family structures are often used instead.
Okay, so let's say you've navigated that, you've built your population, you've got your genotype data for all your markers. What's the next big step?
Constructing the linkage map. This is like creating the road atlas for the genome you're studying.
The atlas. I like that. So it shows the order of markers and the distances between them.
Exactly. A linear arrangement showing marker order and the genetic distances, usually measured in centimorgans, which are estimated from those recombination fractions we talked about earlier.
Okay, walk me through the workflow. How do you actually build this map? What are the steps?
Well, it typically starts with estimating recombination fractions between every possible pair of markers. That's the 2-point or pairwise estimation.
Every single pair. That sounds like a lot of calculations.
It is, especially with modern dense marker sets. Then you perform grouping. Markers that show a recombination fraction significantly less than 0.5, meaning they're recombining less often than pure chance, are grouped together.
And if A is linked to B and B is linked to C.
Then A, B, and C probably belong to the same linkage group, the same chromosome. It relies on that transitive property.
Okay, grouping makes sense. But how do you decide if they're really linked? Or if that low recombination fraction is just, random noise? You need some statistical tests, right?
Absolutely. That's where the LOD score comes in. It's a fundamental statistic in linkage mapping.
LOD score. What does that actually tell us?
Think of it as a measure of confidence. LOD stands for logarithm of the odds. It compares the likelihood of the data if the markers are linked, with some estimated recombination rate less than 0.5, versus the likelihood if they are not linked, the Mel hypothesis, where R equals 0.5, meaning independent assortment.
Logarithm of the odds, okay.
Yeah.
And there's a threshold.
Yes. The standard threshold, widely accepted, is a LOD score of 3.
And what does a LOD of 3 signify?
It means the odds in favor of linkage are 133, or $1,000 to 1. So the hypothesis that the markers are linked is 1,000 times more likely than the hypothesis that they're unlinked, given the observed data.
Wow, okay. 1,000 times more likely. That feels pretty definitive.
It's a strong indicator, yes, that gives us our linkage groups. But then comes arguably the hardest part computationally, ordering the markers within each group.
Right, putting them in the correct sequence along the chromosome. Why is that so hard?
Because you're trying to find the marker order that requires the minimum number of crossover events, or recombinations, to explain the genotype data you observed in your population.
Minimizing crossovers. This sounds familiar. Isn't this related to a famous problem in computer science?
It is. It's mathematically analogous to the traveling salesman problem, the TSP.
Ah, finding the shortest route to visit all cities just once?
Exactly. Here, the cities are the markers, and the distance is related to the recombination frequency, or the number of required crossovers. You want the shortest, most parsimonious path or order.
And TSK is famously difficult to solve optimally, right?
Extremely difficult. It's what we call NP hard.
Yeah.
The number of possible orders for null markers is null factorial divided by 2.
Which gets astronomically large very fast.
Unbelievably fast.
Yeah.
The source material mentions that for just 20 markers, a brute force check of all orders would take a powerful computer over 700 years.
And 25 markers.
Billions of years. It's computationally impossible to check every single order once you have more than a handful of markers.
So brute force is out. How do
Right.
they actually solve it then? There must be shortcuts or smarter algorithms.
There are. We have to rely on sophisticated algorithms, like branch and bound methods, or heuristics like simulated annealing, that try to find a very good, if not always the absolute perfect, order much more efficiently.
Okay, algorithms help. But is there also a better way to use the data itself beyond just pairs of markers?
Yes, and that's crucial. Modern mapping relies heavily on multi-point analysis.
Multi-point, so looking at more than 2 markers at once.
Exactly. Instead of just pairwise estimates, multi-point analysis considers the data from all markers across a chromosome segment, or even the whole linkage group simultaneously.
How does that help with ordering and distance accuracy?
It uses the information much more efficiently. It often employs powerful statistical models like hidden Markov models or HMMs.
HMNs like used in speech recognition.
Similar underlying principles, yes.
Yeah.
Here, the HMM models the hidden state, the true sequence of crossovers along the chromosome, based on the observed state, which is the marker genotype data. It gives much more robust estimates of order and distance.
Okay, so complex stats and algorithms get us the accurate map. The atlas is built. Now, the final payoff, actually finding the genes controlling the trait, the QTL mapping part.
Right. Now, we use the map to find the locations on the chromosomes that influence the quantitative trait we care about, the quantitative trait loci, or QTLs.
And the goal isn't just finding one, right? It's about understanding the whole picture.
Exactly. The goal is to map the genetic architecture of the trait. That means estimating how many QTLs are there, where are they located on the map, how large is their effect, and crucially, how do they interact?
Interactions, like epistasis, where one gene affects another, or genes interacting with the environment.
Precisely. And also things like pleiotropy, where one gene might affect multiple traits. We want the full picture.
So how do we find these QTLs? What are the methods?
Well, the methods have evolved quite a bit. The earliest and simplest approach was single marker analysis, or SMA.
Single marker analysis, how did that work?
It's pretty straightforward, actually. You basically do a simple statistical test, like a t-test or ANOVA, for each marker individually.
So you take one marker, divide your population into groups based on their genotype at that marker, like AA versus AA versus A, and then you just compare the average phenotype, say average plant height, between those genotype groups.
That's essentially it. If there's a significant difference in height between the groups for a particular marker, you infer that there's likely a QTL influencing height located near that marker.
Seems simple enough, easy to do, doesn't even need a full map, just marker data.
Right, those are the pros. Easy, quick, no map needed.
But there have to be downsides, otherwise we wouldn't have needed newer methods. What were the big flaws?
SMA has some pretty major limitations. First, it can't distinguish between a single QTL near the marker and maybe multiple QTLs all linked together in that region. It just sees a general association.
So poor resolution.
Very poor. Also, because you're testing at the marker's position, not necessarily the QTL's true position, it tends to underestimate the actual effect size of the QTL. And related to that, its statistical power to detect QTLs is generally lower because of that location bias.
Okay, so SMA was a starting point, but flawed. What came next? How did they improve it?
The next big leap was interval mapping, or IM. This is where having that linkage map becomes absolutely essential.
Interval mapping, so it looks between the markers now.
Exactly. Instead of just testing at the known marker locations, IM systematically scans the intervals between markers along the linkage map. It basically tests for the presence of a QTL at many positions, maybe every 1 or 2 centimorgans within each interval.
Okay, that sounds more precise. But how does it test a location where you don't have a marker? How do you know the QTL genotype there?
That's the clever part. You don't know the QTL genotype directly, so it's treated as missing data. IM uses a statistical framework called a mixture model.
Mixture model, meaning?
It assumes that the overall distribution of the trait, like height in the population, is actually a mixture of distributions, one for each possible QTL genotype, like QQQQQQ. It then uses the information from the known genotypes at the flanking markers, the markers on either side of the interval being tested, to calculate the probability that an individual belongs to each QTL genotype class at that specific test location.
Wow. Okay, so it's using the neighbors to infer the probability of the genotype in the middle.
Precisely. It's using the linkage map information and recombination frequencies to make probabilistic inferences about the QTL genotypes within the interval.
And the advantages of IM over SMA.
Huge advantages. Because you're testing closer to the actual potential QTL location, you get much more accurate, less biased estimates of the QTL position. You also get better estimates of the QTL effect size. And generally, it has much higher statistical power to detect QTLs.
Big step forward. But IM wasn't perfect either, was it? I remember reading about ghost QTLs.
Ah, yes, the ghosts. IM still had a significant issue. If there was another QTL located somewhere else on the same chromosome outside the interval currently being tested,
Yeah.
IM didn't account for its effect. So the genetic variation caused by that other QTL could inflate the test statistic in the interval you were looking at, sometimes leading to false positive peaks, the ghost QTLs, or distorted estimates of the real QTL's position and effect.
Okay, so you needed a way to isolate the effect in the target interval from the noise caused by other QTLs on the same chromosome.
Exactly, which led to the development of composite interval mapping, or CIM.
Composite, so combining interval mapping with something else.
Right, CIM performs interval mapping just like IM, testing for a QTL within a specific interval.
Right.
But crucially, it simultaneously includes other selected markers from elsewhere on the genome, particularly from the same linkage group, as cofactors in the statistical model.
Cofactors, like control variables in a regression.
That's a great way to think about it. These cofactor markers act as proxies for other QTLs outside the test interval. By including them in the model, CIM statistically controls for the genetic variation caused by those other QTLs.
Ah, so it filters out the background noise from linked QTLs.
Precisely. It helps to absorb the effects of those other QTLs, allowing for a clearer view of the QTL within the test interval. This significantly reduces bias, increases precision, and largely eliminates the problem of ghost QTLs.
Okay, CIM sounds pretty sophisticated. Is that the end of the line, or did things get even more complex?
They pushed it further. The next level, often considered the most comprehensive approach among these classical methods, is multiple interval mapping, or MIME.
Multiple interval mapping. So now we're looking for several QTLs at once.
Yes. Instead of testing one interval at a time while controlling for others, like CIM, MIME attempts to build a model that includes multiple QTLs simultaneously across the entire genome, or at least across multiple linkage groups.
And it can model their interactions, too, like epistasis.
That's one of the key strengths of MIME. It allows you to explicitly search for and model not only the main effects of multiple QTLs, but also the pairwise interactions between them, the epistasis effects. This gets us much closer to understanding the true complex genetic architecture of many traits.
So looking back at this progression, SMA was simple but flawed. IM used the map to look backward markers but ignored other QTLs. CIM improved on IM by adding cofactors to control for other QTLs. And MIME tries to model multiple QTLs and their interactions all at once.
That's a perfect summary. There's a clear increase in model complexity, statistical sophistication, and generally statistical power as you move from SMA to IM to CIM to MIME. Each step was designed to overcome limitations of the previous one.
It really showcases the iterative nature of science, identifying a problem, then developing a more refined tool to address it.
Absolutely. And this entire pipeline from designing the right population, genotyping, building that accurate linkage map through complex computation, and then applying these increasingly sophisticated QTL mapping methods like CIM or MIM. That whole process forms the essential toolkit for modern plant and animal breeders, and for geneticists studying complex traits. It lays the foundation for deeper insights into things like pleiotropy and epistasis.
What really stands out to me is that jump from simple ideas like just comparing means at markers, SMA, to needing things like hidden Markov models for ordering and these complex mixture models for interval mapping. Scientists had to bring in some heavy-duty statistics and computation to make progress.
Definitely. It highlights how progress in genetics is often tightly coupled with advances in statistical methodology and computational power. Biology poses the question, but you often need math and computation to find the answer.
So thinking about that computational challenge, we heard that ordering just 25 markers by brute force would take billions of years.
Computationally infeasible.
With genomes getting sequenced faster, marker density exploding, the data sets are only getting bigger and more complex. How do you see something like artificial intelligence impacting these fundamental algorithms, like the TSP-related ones or the QTL mapping models, going forward? Will AI find shortcuts that methods like branch and bound can't?
That's a fascinating question, and probably where the cutting edge is heading. Can AI learn patterns or develop novel optimization strategies for map construction or multi-QTL modeling that outperform current algorithms, especially on these massive datasets? Certainly a key area to watch. That's the challenge for the next wave of research,